In glycolysis, the glucose molecule is broken into two 3-carbon sugars, which will cause 2 ATP. This 3-carbon sugar is known as pyruvate, and it is formed outside of the mitochondria. First, the glucose needs to become phosphorylized. This is important because it allows the glucose to break down and also creates 2 ATP from 1 pyruvate. This is known as substrate-level phosphorylation. In this process, an enzyme breaks the phosphate off the pyruvate and then bonded with ADP to create ATP. This does not create a large amount of energy, however, it will create a little. During this process, NAD+ will take electrons from pyruvate so that electrons can later be used in the electron transport chain. The NAD+ will then become NADH when it has electrons. After glycolysis, the pyruvate will enter the mitochondria and begin formation of acetyl-CoA. First, one of the carbons will break off the 3-carbon pyruvate and then create carbon dioxide. Then an NAD+ will take electrons off the 2-carbon fragment and become NADH. Finally, coenzyme A will bond to the 2-carbon fragment, creating acetyl-CoA, which has high potential energy and is used to transfer the acetyl group to a molecule in the Krebs cycle, which is an exergonic reaction. After the formation of acetyl-CoA, coenzyme A will be removed, and the 2-carbon fragment will bond with a 4-carbon fragment to create the starting point for the Krebs cycle. Now, the 6-carbon fragment will lose 2 carbons, which will become the carbon dioxide we exhale. Additionally, electron carriers will strip electrons from the 4-carbon which was created after the loss of 2 carbons. These electron carriers include NADH and FADH2, which before holding electrons were NADH and FAD+. The citric acid cycle, or Krebs cycle, will also create 2 ATP for each molecule of glucose. And once the electrons have been stripped, the 4-carbon fragment will reconnect with the 2-carbon fragment, and the cycle will start again.
The electron transport chain is a collection of complexes consisting of multiple proteins with electron carriers embedded in the inner membrane of the mitochondrion, which is folded into cristae to increase surface area. During this process, electron carriers alternate between reduced and oxidized states as they accept and lose electrons. Electrons are transferred from NADH to the first molecule of the electron transport chain, called flavoprotein. Next, flavoprotein passes its electrons to an iron-sulfur protein, returning to its oxidized form. The iron-sulfur protein then passes the electrons to a compound called coenzyme Q, a small hydrophobic electron carrier that is mobile within the membrane. Meanwhile, hydrogen ions are pumped against the proton gradient and into the intermembrane space. The last cytochrome of the chain then passes its electrons to oxygen. With the use of 2 electrons, each oxygen atom binds to a pair of hydrogen ions from the aqueous solution, resulting in H2O as a byproduct. During chemiosmosis, the protons flow back down the gradient through a turbine-like protein called ATP synthase, which spans the membrane. ATP synthase harnesses this force to drive the phosphorylation of ADP, forming ATP. Together, the electron transport chain and chemiosmosis make up oxidative phosphorylation, releasing 26 to 28 ATP in this final step.
Fermentation starts with the same basic idea of glycolysis. However, in fermentation, there will be no oxygen available. This barrier prevents the same amount of ATP production to occur as in aerobic respiration. The 3-carbon pyruvate will once again be phosphorylized in this system and will create 2 ATP. However, this time electrons will be added to the pyruvate to allow it to become lactate. This is due to the enzyme lactate dehydrogenase binding to the pyruvate molecules. Lactic acid is what builds up in muscles after exercise without oxygen. As you can see, the lack of oxygen in the process only allows for about 1/16 of the amount of ATP to be made as compared to cellular respiration, which shows that cellular respiration is a better process for creating large amounts of energy with an abundance of oxygen.